Hello again, and welcome to our video lecture on section 4.5 on cell communication. Here are our learning objectives. Now let's start with direct contact between our cells. It's really important for cells to function. Examples would be our cells of our immune system, how they distinguish normal cells from unwanted cells, such as ones that are pathogens or microbes within our body. And another example would be our sperm and oocyte, or the female egg. Our egg is unique in that it has the glycocalyx, and it allows for recognition by the sperm cells during fertilization through that glycocalyx. And then we have the example of cellular regrowth that follows injury. Our damaged tissue is replaced by cell division in the epidermis, and we have that cellular contact that prevents overgrowth. Now, when we talk about cell communication, most of it is going to take place through ligands. Ligands are going to be molecules that bind with macromolecules, or can be neurotransmitters from nerve cells and hormones from our endocrine cells. They're going to be important for controlling growth, reproduction, and other cellular processes. And we have three types of receptors that are going to bind ligands. Those three are channel-linked receptors, enzymatic receptors, and G protein-coupled receptors. So let's look at the channel-linked receptors first. They are going to allow ions to pass into and out of our cells. It happens in response to neurotransmitters. So if you look at this closed channel receptor here, we can see that the ligand is approaching to attach here to its special site, and that is going to open up the channel and allow the ion to flow through. This can help initiate electrical changes to that resting membrane potential in muscle and in nerve cells. Now let's look at enzymatic receptors. This would be like our protein kinase enzymes, and they have a ligand that will attach to them, and it'll become activated to phosphorylate other enzymes within a cell. So you could see that taking place over here. It really helps to provide a mechanism for altering enzymatic activity. And we're going to see this within our endocrine system. And lastly, we have these G protein-coupled receptors. So you're going to see this process is a little bit more involved. It indirectly activates protein kinase enzymes. So let's take a look at a quick video to see this in animation, and then we'll talk about it in this figure.
Site for a small molecule called a ligand exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
So what I really wanted you to get out of that video is this G protein is made up of three separate subunits, the alpha, beta, and gamma. And so let's put this in our full figure over here. We're going to have a ligand that'll bind to a receptor. The receptor will change its shape and become activated. At that point in time, this G protein will have a GTP molecule bind to it, which activates the G protein. And when that activated G protein travels, that alpha subunit breaks off, and will then activate our effector protein. So we could do this either by this effector protein embedded in the plasma membrane like an ion channel to let ions move through, or the activated protein can attach to an effector protein just here in the cytosol, such as an enzyme. Either one of these effector proteins can then create a secondary messenger, which can be a protein of some sort, and that will continue on to this inactivated protein kinase enzyme that's just chilling here waiting, and the secondary messenger will cause the protein kinase to become activated. That protein kinase will then phosphorylate other enzymes and cause an enzyme to either turn on to cause some reaction to take place or turn off in order to stop a chemical reaction from taking place.